I'd like to invite the first panel to come down and take seats here. And what I'd like to do is first to introduce briefly and ask to introduce themselves the panelists. Sitting directly to my left is Brian Spear. Brian is the Director of Genomic and Proteomic Technologies with Abbott Laboratories. Next to Brian is Marta Gwinn. Marta is a Senior Medical Epidemiologist in the Office of Genetics and Disease Prevention in the National Center for Environmental Health at the Centers for Disease Control and Prevention. She'll, I think, unpack that a little bit more when she introduces herself. And Rex Chisholm, who has introduced himself, is our host and the director of the Center for Genetic Medicine here at Northwestern University. The subjects that we're going to talk about on this panel really are two. First is just kind of definitional. What do we mean by personalized medicine? What do we mean by genetic medicine, genomic medicine? Just trying to make a little bit more sense of the territory and give us some common vocabulary so that as we go through the rest of the day, we have a sense of just what we're talking about. The second is to address some of the scientific issues. I know that as an audience, it's important for you to really understand where people stand. You need to know a little bit more where they sit. Let's see, Marta, did you not get a mic? Oh, okay. Well, yeah, the sound folks will deal with that. As I was saying, it's important for you to know where people sit in order to understand where they stand. And so what I'd like to do is ask each of the panelists first to introduce himself and herself in a little more depth than I did, and to at least give a sense of the issue as you see it. Rex, let me start with you.
All right. Well, I obviously represent the academic and research enterprise in this event. And obviously the goal for many of us is to be able to take the genetics information that comes from a human genome sequence, for example, and extend it to all these wonderful things we keep hearing about as possibilities of the human genome sequencing effort and of personalized medicine. One of the key things that takes, I think, is to be able to actually connect up what the characteristics of all of those genetic variations that make us all different from each other, and how those characteristics actually lead to the diseases that we will suffer in our lives, how we might respond to drugs, how we might respond either well or poorly to drugs, maybe even what the outcomes of therapies might be. And all of this is, in many cases, an interaction between genetic information that we carry in our DNA and the environments that we either find ourselves in or we put ourselves in. And how do we actually make that transition from that one canonical human genome sequence that's in GenBank in NIH is computer banks and convert that into that kind of information? And one of the things that it requires is research in a very large number of individuals where we know what their kinds of genetic variation is, where we know how that genetic variation actually correlates with the diseases that they suffer from. And one of the ways we think we can do that is, for example, through the creation of biobanks, which give us access to genetic information from individuals, gives us access to as broad a picture of their health status as we possibly can. So this involves the use of electronic medical records. It involves the use of a whole series of epidemiological approaches. And it is only through the combination of each and every one of those that I think we're going to actually be able to achieve the goals of personalized medicine. And just to put in a plug and extend Dora Hughes' comments briefly, and I'm sure we'll come back to that later, I think an important issue for all of us on the academic side is to think about the fact that we have this incredible resource available in terms of people's medical records and their health status, And how do we find a way to be able to enable us to use it for the research efforts that we need to use it for without in any way putting at risk the ability of those individuals to receive the health care and have that health care paid for as they move forward. And I think that's a big challenge for us in the area of personalized medicine.
Thanks, Marta?
Yes, well, excuse me, I'm Marta Gwinn. I'm a public health physician, and for the last 7 years or so have been with what has now been renamed to better reflect our mission, the National Office of Public Health Genomics. And what I do there mostly is work with something that we call HugeNet. That stands for Human Genome Epidemiology Network. And it really started out about 7 or 8 years ago as really something that was only a gleam in the eye, but it's an idea that has caught on really faster and faster, not only within the U.S., but especially in international communities where the focus has always been more on community medicine than on the individual. Because, and I think this ties in very well with what you've said, there's almost a paradox in the sense that in order to truly personalize medicine to the individual, you have to start with knowledge of the distribution of genetic, environmental, and other kinds of risk factors in very large numbers of people. So what we're trying to do is bring the tools of the population sciences together with the tools of basic science and basic clinical science to really accomplish the translation that's required to produce health benefits from the public investment in genomic research. So in the same sense that systems biology acknowledges the complexity and the role of stochastic processes in biological systems, we are in a sense taking a systems approach to the relationships among disease risk factors and outcomes within a broad population. And I think it's going to take a large interdisciplinary effort that really melds together these different sciences to make sense of the research output that's now growing. So the main function of HugeNet is basically to apply the tools of epidemiology to a synthesis of information on gene-disease association, gene-environment interactions, and their potential relationship to health outcomes, not only in individuals, but at the population level.
Good. Thank you. Brian?
Thanks. First, just to introduce myself a little bit more. 30 plus years ago, I was on the faculty here at Northwestern, actually up in Evanston. I'm a geneticist and molecular biologist by background. And at the time I was in Evanston, I built and had approved the first recombinant DNA lab at Northwestern, which gives you an idea of the passage of time. But I've been at Abbott Laboratories for 25 years, and while there I've worked in research, mostly molecular biology research. I've worked in our diagnostics business, developing tests and focusing in many ways on molecular testing. And then 9 years ago, I was asked to put together our first effort in pharmacogenomics, and I've headed up our pharmacogenomics group there for some period of time, more recently taking over responsibility for proteomics and genomics generally. From my background, I'll be unabashed in saying that the view I take on things is one of a large commercial pharmaceutical and diagnostic company, and I'm sure that will come through in the statements I make. It is our goal at Abbott and throughout the pharmaceutical industry to be able to bring to the market and bring to patients safe and effective treatments to cure and alleviate disease. And there are a number of ways, and a lot of money gets spent in trying to do this at Abbott and elsewhere. But one of the ways that we are more and more taking advantage of is having a better molecular understanding of the patient, understanding the disease, understanding the patient, understanding how our drugs work, understanding the interplay between identification of molecular changes, disease, and most appropriate treatment. So we use genetics and genomics throughout our processes. We use them to understand our patients better. We use them to interpret some of the data that we get from clinical trials. We use it to design clinical trials, which will either be smaller, faster, more informative, or safer. And we will be using genomics to direct drugs at the most appropriate patients so that we can deliver the best treatment as quickly as possible. So that's my approach to things.
Good. Well, thanks to all of you for those introductions. I'd like to dive in just on that very first definitional question. And at least two of you, it sounds like, spend much of your time and the time of your teams on understanding the molecular, the genetic or genomic basis of human disease. In some cases, trying to reach towards treatments or cures of those diseases. Could you distinguish that whole field of genomics from the more general rubric that we're talking about today of personalized medicine, and try and place genetic medicine or genomic medicine in the context, in what I think is the broader context of personalized medicine? Brian, do you want to jump in?
I knew that would come to me. There really are two different ways to look at it, although the overlap is enormous. One is understanding the molecular basis of disease that lets us identify new points of intervention, particularly for pharmaceuticals. The other is identifying the genetic differences among patients so that we know how to direct medication. Of course, in many cases, those two are the same, and the best example, I get to use it first, is Herceptin and the Hercept test, where breast cancer now in some ways is being looked at as HER2 positive and HER2 negative breast cancer, because the decisions on treatment may change depending on the results of the Hercept test.
HER2 being a genetic variation.
It's a cell surface protein which is overexpressed in some breast cancers and not in others. And the course of the disease is different. So in this case, we can look at the disease in two different categories using a genomic marker, and selection of treatment can be for the two different categories of disease.
Yeah, so it's the idea then of personalization of treatment by selecting the most appropriate pharmaceutical product to fit the genetic profile of the disease in that patient.
That's
right.
Amplify that just a little bit. There's a piece, and Brian alluded to this, that comes before that. And that is actually just fundamentally understanding the biology. The beauty and the excitement of genetics and the fact that we've deciphered DNA is that we have those instructions in our hands. And so now the task for those of us that do basic biomedical research is to really understand what that protein does and how that protein interacts with that protein in the process of a normal biological function, recognizing that it is in fact variants, defects, misuses, the collection of all of those interactions, gene products, whatever, that actually lead to dysfunction at the organ level, dysfunction at the individual level. And so there is a first step in genomics that is simply the deciphering of those instructions and the proteomics piece that derives from that, which says that there's these proteins interact with it, and then even the systems piece that says this is how these things interact when put in this kind of an environment or that kind of an environment. So that's also a piece that's sort of outside the personalized medicine, but which derives from genomics.
Now, 5 years ago or so, when we completed the first draft of the map of the human genome, there was a huge degree of hype, I think, about how quickly we would be able to reap the fruits in terms of personalized treatments of diseases. But if I'm understanding, Rex, what you're saying, that's really only the first step. I mean, that's the map of, I don't know, of Francis Collins or of Craig Venter's genome. And what you're saying is that really we need to understand the range of genetic variation and its impact on biological processes. That seems like a much larger research prospect. And where do we stand now? And can you maybe help sort the hype from the reality, or maybe more say what's the timing with which we'll really be reaping these fruits?
Well, so there's a lot of effort underway right now to try to go that next step. I mean, so there are a couple of features. First, going the next step in terms of just understanding what the range of quote-unquote normal is. How do we all differ in our DNA? You know, the number that people throw out is, out of the 3 billion letters in our DNA, we differ at 1 out of every 1,000. That may be a good number, it may not, depending on how you view the particular piece of DNA you're looking at. But just to understand for the people sitting in this audience, how much variation is there. And some of that leads to disease, and some of it has no consequence whatsoever, and we need to do that. That's the first step. The second step is then to understand how each of the bits of those pieces of DNA that encode something, that make a protein, how they do that. And those are the kinds of projects that are underway right now, to take and ask, in a heart cell, what are the proteins that are there, and how do they change as the heart cell is exposed to different environments? So we are in the midst of those kinds of very large-scale studies right now. And then, of course, all of those fit into the epidemiology side. How does the understanding of that biology now allow us to link to the epidemiological studies that say what the relative population risks are?
Marta, is there anything you'd like to add to that? Maybe put this into more of an epidemiological and public health context?
Sure. Thank you, because that provides a very nice a sequence, I guess you would say, leading up to what you know, of course, we think is the most important, which is looking at population variation in these various traits. And you know, to put it into context, as you said, the entire, you can understand the entire human genome sequence by looking at one genome, you know, one person's genome, effectively. And of course, last year we got results of the initial phase of the HapMap project, which looked at distribution of haplotypes in 4 different populations in the world. And just to give you a frame of reference, if you don't know, that those HapMaps were established based on about 270 people.
Can you say what a haplotype is, please?
A haplotype is a combination of genetic markers that are close enough together that they tend to be inherited together. And those are the patterns that, for example, projects like the National Genographic Project are using to trace your deep ancestry, if you have heard about that. So anyway, that phase, the HapMap project, used about 270 people. Now, one thing that we're doing right now at the CDC is, and this is in collaboration with the National Cancer Institute, is there's a long-standing population survey called the National Health and Nutrition Examination Survey that's been conducted in the U.S. periodically since the 1970s. And one phase of the survey that was conducted in the mid-90s collected DNA samples from about 7,000 people. And these people are a probability sample of the U.S. population from, you know, age 2 up to age 90 plus. And it represents, in a probabilistic way, people from all the different regions and racial and ethnic groups in the U.S. population. And with this DNA bank, with collaboration with NCI, we have now genotyped about 100 genes that have been associated in various types of epidemiologic studies with disease. So we're going to have that. But that's still a very limited, very limited database with which to understand population variation. One of the key, most important concepts in this is that we need to know not just what, you know, variants exist in this sample of people here or in that sample of people there, but in a population-based sample of whatever the population is that we aim to take care of, because only then will you know how many people, what proportion of the cases, you know, what's the likely size of the group of people that could benefit.
In your opening remarks, you talked about stochastic processes. You've just mentioned probability. And I'd like one of you, if you could, just to clarify a little bit, as we get better and better mappings of the genome, of the haplotypes, to explain a little bit just how deterministic is this. Brian, you mentioned HER2/neu, and that's a fairly deterministic marker for that tumor type, as I understand it. There are other examples like chronic myelogenous leukemia that's traceable back to a single genetic variation. Now, is that likely to be the case, that most diseases have a single gene that explains its presence or absence? Or is it a more probabilistic mix, as you mentioned with the haplotype clusters of markers?
Well, I think the conventional answer would be it's quite probabilistic, and there's low penetrance, that is low effect genes, and there's high penetrance genes. And all that is very true. One thing to keep in mind, just to put it in perspective, if you consider 3 attributes, let's say, of mine, pattern baldness, blue eyes, and my political party affiliation, and you were to try to predict from these what my sons are, political party affiliation has the highest inheritance. And that, I think, does not have a genetic component. So there's a lot going on here beyond the simple genetics of it. There are certainly some genes which are highly penetrant, cystic fibrosis, Huntington's disease, and so on. In other cases, the popular belief among scientists is that with common chronic diseases, depression, type 2 diabetes, and so on, that there are genetic determinants that influence whether or not you're likely to have it. I suspect as we get closer and closer, we'll find that the genetic determination is stronger than we currently think, and yet it's going to be dependent on multiple genes. And we would have to then understand the variance in each of the genes and their pattern of interaction to be able to make better predictions. But I think those predictions will be possible. That doesn't hold for political parties.
Rex? Yeah, I'd like to extend that a little bit, because I think that's a very important point. I mean, one of the big changes that we're seeing in terms of the whole concept of genetics and genomics, and why people are so excited, is that what genomics offers us is the opportunity to look at all those variations across an individual, and then to take 1,000 individuals and look at the genetic variation across 1,000 individuals, and begin to dissect out the fact that it's not just inheritance of a single gene that matters, which certainly matters for what we call the Mendelian single gene diseases, but for a lot of reasons, including natural selection processes, those are not the most common diseases that we have. The most common diseases that we have, the ones that Brian mentioned and the ones that all of us will suffer from and have relatives that suffer from, are things which probably result from sort of what I like to call the roulette effect. Our genome has been mixed in certain ways, and we've come up with a collection of 3, 5. Some diseases, it's been estimated there may be 9 or 12 contributions to that disease. And it is sorting those out that is our challenge. And that requires not one individual, but probably hundreds of thousands of individuals.
Let me make an example here just to give an idea, sort of to amplify the approach I suggested and make it clear how difficult this will be. If you think within your own families about inheritance of characteristics, ear shape, voice, sense of humor, things that say, oh, he's got his father's sense of humor. That's probably true, and it probably is based on genetics. Is there a gene for sense of humor? Well, some people lack it, but I think generally there's no single gene for sense of humor. It is a combination of different genes which can be inherited together and can lead to some very subtle but clearly inherited characteristics. That's probably true with disease also.
Marta?
Well, I'd like to go back to your idea of the roulette effect and the the fact that it's probably combinations of multiple genes, which you just described using another metaphor, the inheritance of the sense of humor. And that is that, you know, it's true that if the more information you have about something that you want to predict, the better your prediction is likely to be. But if you're talking about predicting something that's very common, like heart disease, remember that each of these combinations is going to be quite uncommon, because each genetic variant is also inherited on a probabilistic basis. So once you start multiplying those probabilities, any one combination is going to be quite rare. So it seems quite possible that at least where common diseases are concerned, we're going to be looking at combinations of combinations and multiple types of combinations. And so it really does become quite complex, and it really does still remain a probabilistic process. I mean, I can think of this, especially in terms of prediction, you know, where obviously from a public health standpoint, we're very interested. You know, it's almost similar to weather forecasting, because, you know, when you have someone with cancer and you're able to actually measure markers on tumor cells and so forth, you have really zeroed in on the weather forecast for this afternoon. You have a lot of information that's very proximal to the disease process and also to the therapeutic intervention. But when we're talking about, you know, starting in early childhood to try to prevent heart disease, then we are trying to make very long-range forecasts, and those are highly, you know, unreliable for the most part. So it's only by making more sophisticated models, as they are now in climatology, that use information from multiple different sources as well as historical patterns, that we can really hope to have any better prediction. So basically, the closer you are to the disease endpoint, you know, the better your predictions are going to be. And I think this is where, like in pharmacogenomics for cancer specifically, some of the earliest successes are going to occur. And it's only as we learn more and more that we're going to be able to intervene earlier. And the further upstream you are from disease, the more difficult it is.
That's very helpful. And if I take that metaphor of weather forecasting and also just weather detection, one of the ways that we've increased the power of weather detection is increasing the number of sensors, of places around the planet from which we gather information. And then, of course, improving the models and increasing the computing power to be able to deal with all of that information. I think I've just heard you say the same thing about increasing the predictive power of genetic and other biomarkers, that if we move from 2 people for the map of the human genome to 270 for the map of haplotypes, to beyond, and this is really where my question is, what sort of scale of research is needed, and is that research in the pipeline now, you know, without getting to the usual plea for more money for biomedical research, which you know, is maybe an inevitable outcome of a conversation like this. What's coming down the pike in terms of research, and how large a study is sufficient? How big is enough?
Well, I can just make a couple of comments on that. So one example of an initiative whose goal is to do this is the BioBank UK. This is an initiative where they're enrolling 500,000 residents of Great Britain in a study to actually begin to collect the DNA and link it to their health status in a way that would allow us to have that kind of information for up to 500,000 individuals. The BioBank UK and the New Gene project that we have here at Northwestern are examples of that. Our target is 100,000 individuals. Have formed a consortium called P3G. It's a public population projects in genomics, and actually the CDC is a very active participant in that as well. And the estimate from that group is that we really need to have on the order of 2 million individuals and have their genetic information and their health status together in order to really make a significant statistical inroad into that. So that's one starting number. We need about 2 million. And what we think we probably need are, like HugeNet, national networks, international networks of collaborative groups, which raises a whole interesting additional set of problems. Like we need to make sure we collect on our data in a way that we can share it and that it's interoperable, if you will. And so that's a really big challenge for us going forward.
In research, like in most things, more money is better than less money. And more samples is better than fewer samples. But the number you need really is going to be determined by the question you want to ask. In order to have a bank where you can ask any question, I think 2 million is the number we'd be looking at. But in many cases, we're interested in a specific question. We're interested in the genetics of major depression, trying to determine if we can identify a gene that will enable us to see a new intervention point for depression. And in that study, we had access to 1,200 individuals in 110 families. And we're able to find a new gene that we're now investigating for potential new antidepressant medication. In other instances, for instance, trying to understand some of the rare but very specific adverse drug effects that come from drug treatment, if you have 30, 40, 50 individuals who have suffered a particular characteristic adverse side effect of drugs, that should be enough that we'll be able to identify a gene that is responsible for it, if that's the case. So it's going to range over a wide difference in numbers, but it's all dependent on what you want to know, because that's going to determine how many individuals you need to look at with the characteristics and how many people are the non-affected controls.
Yeah, so it sounds like what I'm hearing here is that the narrower the question you ask, the smaller the population needed, the more simple the genetic mechanism, the fewer genes involved, the smaller potentially the population. The more you're able to be selective of the population that you're researching, taking your example of people who already have suffered a rare adverse drug event, then the smaller the numbers.
You left out one part, which
yes.
is luck.
Yes.
And I don't mean this just trivially. Absolutely. Because, yes, it's true, the stronger the genetic effect, or the fewer number of genes involved, the fewer samples you need. But you don't know that until you've done the experiment.
Right.
And that's actually the beauty of having the very large collections, because if we could get to the point of actually having the sequence of 2 million individuals, then we would really be able to start to think about tackling, it's this example you gave, of having multiple sensors around the world sensing the weather. Then we would really have the maximal number of sensors possible.
Yes. Marta, I'd like to come to you and ask you to bridge this back to a population health, public health point of view, because I think much of what we've been talking about here really has it maybe has been a little bit implicit, but the model has been, how do we identify the right treatments, the safest treatments for an individual, and get those treatments to that person. And you know, what we've said is, gee, you know, it's so complex that really, you know, if you've seen one person with one disease, you've seen one person with one disease, but you don't do business in that. You do business in populations. Can you help reconcile that for us?
Yes, well, we actually think that the advent of genomic research and thinking, especially because it has sort of captivated the interest of the public as well as the scientific community, this really provides a good opportunity to try to bridge the gap between what has unfortunately diverged in the United States over the last 100 years or so, namely the fields of medicine and public health. Because we see genomics as a good way to bridge the traditional medical approach of, you know, one patient at a time, with the public health approach, which is, you know, mass level interventions, to something that's in between. So we don't have to go with either, you know, one at a time or one size fits all, but to try to bridge that approach by defining in an evidence-based way groups that can benefit from different types of interventions. And of course, I'm thinking really further upstream along the lines of prevention, but you know, one tool that we are using, and our keynote speaker, Dora Hughes, actually mentioned this, is family history. And we like to say family history is very genomic. I mean, you said so yourself, the sense of humor. Family history is very genomic because it captures not only genetic inheritance, but shared environments, shared cultures, shared behaviors, and importantly also captures combinations of genes that are not measured, but that have been expressed as disease within a family. So could public health take an approach that falls somewhere between the one at a time and the one size fits all by identifying high-risk groups defined by family history? So this is the basis of a research project that our group is doing, and actually Northwestern is one of the participating groups of networks of primary care clinics. There are 3 different ones. And we hope to be able to show the value of this approach.
Mm-hmm. I'm afraid I've been monopolizing our panelists a little bit at all of your expense. Let me ask the mic runners to stand, and if there are questions or comments from the audience, I welcome those now. Yes, right here in the middle.
Promise of personalized medicine. And in fact, that's what has been driving a lot of the Human Genome Project. But my concern, though, is that, you know, while we have these interesting and very exciting stories like HER2 and how we can identify sort of the best treatment for that underlie particular pathways, we also have fiascos like BiDil, where many underserved, or in particular, you know, with the BiDil situation, where it was the African American population that was sort of identified as a group that had similar sort of similar pathology, similar disease history, similar genotypes which impact health. And so the focus there was to develop a drug that would sort of fit the group. And my concern is that as we talk about personalized medicine, in particular genomic medicine, there will be situations where many of the drug companies will sort of drop the ball, like they have with BiDil. And instead of taking it to the extent of the individual, they then say, okay, well, this group, since we're trying to make money here, obviously, we're going to market this to a group instead of an individual. The ball is then dropped. And so I wanted to hear sort of how you guys felt about some of these issues. I think these are important issues because if you think about it, as we go towards personalized medicine, we sort of have to have a paradigm shift in terms of how drugs are marketed to the public. And I think that that's not sort of, you know, it hasn't been addressed yet.
Brian, you're the natural person to take at least a first answer to that question.
Yeah, BiDil is an interesting case. It's a antihypertensive, as I recall, that when tested in what's called a typical clinical trial population, which intends to be reflective of the population as a whole, turned out not to be more effective than conventional therapy, and the drug was dropped. It was later on tested in African Americans based on the reinterpretation of the initial results, and it turns out to be effective, or more effective than the conventional therapy in African Americans, and it's now being, I think it's being reviewed for approval right now.
It's been approved.
It has been approved. Well, this presents a challenge of many different sorts, and one that I don't think the pharmaceutical industry is yet clear in terms of how it's going to be addressed. From my standpoint, looking at the genetics, my concern is to be able to identify whether individuals are more or less likely to respond to a drug, rather than characterizing them as Asian, African American, Latino, or whatever, because within each of those groups, there's going to be a great variability in how one responds. So I would hope over the long run, we can ask questions about the individual, have best treatment for the individual, irrespective of any of the sort of initial signs, symptoms, or indications that a physician would encounter when first meeting your patient. We're a long way from that, though.
Marta?
You started your comments by referring to the BiDil story as a fiasco, I think. And I agree. I'm not sure if I feel that way for the same reason that you do, but here's where I think, you know, a big mistake was made in the BiDil story. And that is that in the initial trial, there was no finding of benefit overall. And so at some point, someone got into the original data and decided to look at subgroups, and they sliced it this way and sliced it that way, and found what they thought was perhaps a beneficial effect in the trial participants who were African American. The next thing that happened was a trial was conducted in African Americans only, and that is the basis of the licensed drug now. This is not the right way to do things, and we call this race medicine. This is not the right approach, because that was a subgroup finding that really needed to be evaluated in another trial if it was going to be evaluated at all. Now, there are interesting aspects of this story that include the non-scientific and non-medical, and I'm not going to get into those, but I will say that given that this has happened and that the drug has been licensed, there are more than a few people who are quite eager to replicate this scenario and have already zeroed in, including Decode Iceland, for example, which there are not too many African American people living in Iceland, but they are very good at identifying, you know, commercial opportunities, and so have partnered with American researchers to do additional trials, and they have another cardiac drug in the pipeline that they want to market only to African Americans.
For those who don't know Decode Iceland, can you say just a couple of words about what they've done there?
Well, Iceland is a very unique country in that almost all the people who live there are descended from about 900 Vikings who settled there some centuries ago, having picked up Irish brides on the way. And so they have maintained very, very, very detailed genealogical records there, and so are able to establish really incredible pedigrees going back many, many, many generations. And this provides really a wonderful basis for doing the kind of family-based gene discovery research. And they've taken sort of a public-private approach where an entrepreneurial imperative embodied in the Decode Iceland company has signed up, you know, a good proportion of the people who live there to participate in a very large biobank.
So a large biobank combined with the genealogical pedigree information, and then commercialized. Let's take another question. Yes, right here.
Just a little bit further on his question about the paradigm shift from the drug company's point of view. You now look for the blockbusters. What you're going to eventually look for is, instead of 3 or 4 blockbusters a year, you're going to have 20 orphan drugs a year, because you're going to have subpopulations of subpopulations that a particular drug is going to be good for. Not any drug is going to be good for everybody with depression. It's going to be a subpopulation of the depression. So your paradigm shift is, how do you deal with that when for so many years you only looked for the blockbuster and needed incentives to look for anything that's sold to less than umpteen million people? And that's the paradigm shift I think you need.
That is a very clear logical assumption, and I don't agree with it. That is, it's not so much that I think that what you said was wrong, but that I think depicting where the drug companies are now and where we will be is not necessarily correct. Right now, if you treat patients with an antihypertensive and they turn out to be non-responsive, they will be non-responsive whether or not you have a genetic test. And by and large, if blood pressure is not going down, there will be either change in dose or change in treatment. And so to some degree, we are now already sorting the drugs out according to when they work and when they're tolerated. In this case, it's a slow, very inaccurate, single patient trial and error process. So at the first level, I would say the advantage to genomic testing to associate with selection of therapy is going to simply get you to the right one earlier, rather than changing the actual market size. At a deeper level, however, and this is the researcher speaking now, I think that by understanding the molecular basis of disease and the molecular basis of response or response failure, or the molecular basis of adverse drug reactions, it may allow us to develop drugs which really do work across the spectrum of the indication in the drug label. So that we can now, in choosing between drug candidates, one that works for a subset of patients and one that works for all of the patients, be more and more able to go to the one that works in all of the patients. So I think that you're correct that there are some instances where we will take a disease and categorize it into smaller and smaller groups. Some of those may turn out to be orphan drug categories. But I think by and large, we're still going to be looking for drugs that are effective across a wide range of people. And I mean truly effective, not just indicated for.
I think there's, if I can just make one more comment, I think there's another advantage to pharmaceutical companies of this approach that often isn't discussed, and that is sort of on the liability side. If you can actually better say that this is the person that will be effectively treated by this drug, you're indeed reducing the number of people that you're giving that drug to, but you're only reducing the people that aren't going to benefit from it anyway. And what you do as a consequence of that is you also reduce your liability, because those are people that are at least equally likely and perhaps more likely to have an adverse side effect to that drug. So I think that on the liability side, there's also a benefit. So I understand the concern. The concern is, will there really be effort on the commercialization side to bring to market these exciting new drugs that address a fairly small and focused population? And I think it remains to be seen how effectively we do that. But I think there are also many other components to that that may be positives for companies that bring it out. And it may not be a large pharmaceutical company that brings out those kinds of drugs, but it's a market opportunity for a smaller company that could also still be very profitable in that arena. So I think what we need to think about is the paradigm shift in terms of how do we affect commercialization of our knowledge. And I think there's lots of models out there and lots of opportunity. And the one thing that the marketplace is very good at is where there's an opportunity to allow it to be pursued.
Yes, I'll also say that our next panel will deal substantially with just the question, Rex, that you posed. There was a hand up way in the back there, yes. Can you hold your hand up so the mic runner can see you?
Hi, my name is Jim Caput, and just for quick background, I have a company called Nutrigenomics. It's not a company that believes in genetic testing for nutrition, because we're not ready. But I'm also a consultant, academic consultant at UC Davis, National Center for Minority Health and Health Disparities. I'm at the University of Illinois, Chicago, and I also consult with the Nutrigenomics organization in Europe, NUGO. that's just background, because what I've heard only a little bit from Marta is environment matters a great deal. And if we're going to go on a personal path or a path to personalized medicine, we have to consider environment more. And if, in Rex's example, 2 million people is not only 2 million different SNPs, or individuals with 2 million different SNPs, they have 2 million different environments, different cultures, and different genetic admixtures. I mean, ultimately, we look at admixture as like an African American population, which is recent, but when you look at genetic history, we're all admixed to some degree. Some of my genes have come from chromosome and from Africa and have not changed. So when you talk about 2 million people, you're again averaging out across all of those people. And so we have to think about a different way of designing the experiments to include environment. And so far, that hasn't been done a lot, let's put it mildly. And then the only other point I would like to make is that we're defining personalized nutrition as people with disease states. And I would like to ask the question, why don't we include in personalized medicine prevention? Because our healthcare costs are just going to go up if we design better and better drugs for groups, whereas we can reduce healthcare costs if we take the information from genetics and nutrigenomics and pharmacogenomics and apply it earlier in life. Comment, and then I would like some comments or as a question about if we can extend personalized medicine into prevention.
Yeah, actually, thank you for that comment. That's excellent. I think it's a wonderful broadening of the scope of the conversation in a very useful and productive way. Panel, I'd like to frame it into a closing question, which is to ask you to put your forecaster hat on and think out 5 years from now. So we're nearing the end of 2006. So as we're nearing the end of 2011, I'd like you to talk about what you think the state of personalized medicine will be at that time, taking into account that gentleman's fine comments of the extent to which we'll understand the interactions of environment. Let me add behavior to the question as well, environment, behavior, and genetic endowment, and how to the average citizen in the United States will this present itself in terms of relation with physician, relation with other entities of public health, that mediate their environment, their food, and so forth. How will this knowledge be applied? Right?
Well, I think in 5 years we're going to see not a major shift in the penetration of personalized medicine in its appreciation and uptake by the medical community generally. I think what we will see are individual products, whether tests or pharmaceuticals linked to tests, that will become more and more frequent in the formulary or whatever selection of medications are available. We will see more of them in cancer. Cancer is still going to be leading in large part because what we test for is also what we treat with the drug. So there is a direct link. I think that we will see drug companies introducing new drugs which are tied to a test which is going to be part of the indication for the drug. We'll see those appearing over 3, 4, 5 years. I think the areas, again, cancer, I think we'll see some significant changes in the neuropsychiatric area, in large part because there's such a need for better ways to characterize patients, and because there are some fairly pronounced genetic effects among diseases. And I think we'll also see more and more tests available, either a licensed test kit or a laboratory test, which will indicate people who are at risk for one or another adverse events from drugs like the warfarin example, which is appearing now, and a few others. But I don't see a major change. I think it will look a lot like it looks now, except a little bit more of it.
Mm-hmm. Marta?
I was kind of hoping you would ask him next, because I'm kind of at the opposite end of the spectrum, maybe. But, well, what we'd really like to see in the next 5 years is sort of a transformation in the way we manage knowledge, so that we have as much knowledge integration as we have knowledge generation. Because one of the problems we have right now is it's almost like drinking from a fire hose. There's tremendous amount of research that's pouring forth, not only in the United States, but around the world on, for example, associations of genes with various intermediate health states or diseases, gene-environment interaction studies, and even evaluation of potential genetic tests. And yet it's very, very hard to bring order to this body of knowledge because, among other reasons, there's lack of standardization in measurement of environmental factors and measurement of phenotype. I like to annoy geneticists, of which I'm not one, by saying that's the easy part. That is very easy, measuring a SNP in someone, as opposed to really evaluating, you know, a person's nutritional environment even, for example. So one of the things we're trying to do with HugeNet is really, and also the EGAP project, which was mentioned earlier in the keynote, is really to try to bring some order to the research that's already out there. This is relatively low cost compared to all the money that's going into actually generating the research results themselves. And through engagement and collaboration and I guess I would even say proselytizing, we're trying to bring together through initiatives like the P3G, people who perhaps are in different disciplines but have the same interests in mind, namely bringing to the public genetic tests, gene-based interventions, and above all, better preventive interventions that can truly improve the health of the public, because that's really where I think the public enthusiasm for genomic research lies. It's like, it's very much, what is this going to do for me to keep me and my family from getting sick? So in the next 5 years, to get back to your question, what we really want to have is a much more integrated management of the knowledge that's generated by basic and population-based research, harmonization of the data, and interdisciplinary decision-making that includes not only the basic sciences and the clinical sciences, but the applied health sciences and public health, epidemiology, as well as social sciences. You know, the determinants of disease that are environmental and social are the bread and butter of epidemiology. So we're coming at it from the opposite direction. You know, and in some cases, it's a tough sell to the public health community that yes, you do have to take inherited factors into account. But the fact is, we've got to make a better attempt at synthesis across all of these domains that affect human health.
Rex?
Well, so I think we are really in a stage where we're in the early stages of an exponential growth. And where we are today is we're in the research phase of that. We are acquiring all of this data. And you know, as a geneticist, it doesn't perturb me to hear that phenotype is hard to capture because we know that. That's one of the big things that's a real challenge. I just returned from a 3-day meeting where we were focusing on how does one think about describing a phenotype in a way that can be captured in a computer and linked to those genetic variations. And I think there's a whole range of research opportunities, both in terms of understanding what those genetic variations are, how they contribute to gene effects, how they to drug effects, and how they contribute to disease. But there's also room for an important room for databases and computation and all of the information sciences and better statistical approaches. All of these are going to be required. And as we put more and more of those onto this problem, what will happen is every year, instead of just being today where there might be already a dozen drugs where you can actually do a test to determine whether someone will be responsive or non-responsive, next year it'll be 2 dozen, and the year after that it'll be 4 dozen. And so I'm not sure what the number is 5 years from now, but it's going to be quite a few more than today, and we'll be on a trajectory that will actually take us towards where we are. Will you go to your doctor and hand them your DNA sequence and they'll say, here is a collection of drugs that you'll take for the rest of your life? That's, I think, a fantasy. But the idea that we will better be able to, every year, do a better and better job is really what we're all about.
Yeah, so a wider armamentarium of drugs, some of which right now are characterized as not having enough responders to merit marketing, but then in 5 years we'll begin to make them available, and many more following that. Well, thank you very much.